questions from the crowd. Maybe I can start with some question. I guess maybe for Mo, maybe for Shai, but you know, from in looking at the field of transcriptomics, which actually we've been doing a lot of work on gene expression over the years, but personally I've only worked on bulk RNA sequencing data and not so much on single cell sequencing data. I've been following that field with great interest in the past several years, and my question I'm interested in both of your thoughts on is how much proven value did we get from going to the resolution of single cell data as opposed to the bulk RNA sequencing data. And I ask this also because of what you showed, Shai, where basically you looked, this was on bulk RNA sequencing, I believe, but still you went from the resolution of 20,000 genes and you said, actually, this is too much for us. And we didn't get information when we worked at that level, and we went up a level, and when we used the higher resolution gene expression data to look at a lower resolution cell type data, then we made more sense and we got predictive value and so on. So in that case, you didn't even need the molecular data. If somebody gave you the technology to measure cell type distributions, it seemed like you would do a better job than the molecular data. So I'm wondering your thoughts on, and you've done a lot of work on single cell data, and there's methods I know that decompose the bulk RNA into cell types. So yeah, wonder your thoughts on this issue.
Now? Yes. I'll start because it's going to be extreme, so I'll let Mo recover afterwards. So as I said in my talk, I think, and it's one of the biggest revelations, hidden secrets of genomics is that gene expression is an expensive, confusing way to count cells. And I say this in two ways. So one is it's confusing because you're getting, tens of thousands of genes and you're trying to make sense, and it's just difficult because the unit you're trying to understand is in cell space. But the other side of this is if you look at the information that's coming out of single cell data, it's basically, so number of differentially expressed genes, if I zero into a particular cell type between two conditions is very small. We're very used to, if we're looking at the RNA-seq world, I have thousands of differentially expressed genes in my data, right? 90% of them can be explained by cell composition. We've done this now in thousands of data sets where we basically took the bulk gene expression data, deconvolved the cells, enumerated what are the cells in each sample, and then just regressed out the cell composition. 90% of differentially expressed genes can be explained by the cell composition. Even interestingly is the 10% that remain make up only half of the differentially expressed genes in the data. The other half were hidden by the cell composition, right? So if you take that idea that on one hand, really the bulk data is giving you cell type information for the most part, and then the single cell data, when you look at the particular cell type, really there aren't that many differentially expressed genes. It basically tells me that if our unit of exchange was a cell state, right, we basically would have been in a much more efficient way. I wouldn't need many data points to describe this here. I'll just say, oh, this is an inflammatory monocyte that expresses CD16. You would get what it means. We have a little dictionary, CD16 monocytes secrete this and this, and we'd be done. That's my personal view on this. Add to this the technological piece, which is that single cell data at present, though amazing advances, and Mo mentioned this in his talk, no low abundance information, where the most interesting genes in the genome, and I think you showed this back in 2003, are genes that are low abundance regulatory transcription factors. They're missing. You can hunt for Th17 cells in single cell data, you will not find them because IL-17 is expressed low, and so it goes on and on. I think the answer, at least the one I've taken to this, is it's about how data types complement one another. Ultimately, we want to build an understanding of biology that isn't tethered to any single data type measurements. It's tethered to biological concepts that we model, and where the data came from, well, we're going to algorithmically put pieces together to make sense. So quite extreme, but I'll let maybe Mo give the positive side of this.
Yeah, I fully agree actually with Shaya on this, that this has to be always analyzed in the context and in the type of like the experiment and also the disease model and the context that we're trying to infer conclusions here. That in most cases, so in my experience, in most cases, if you want to, like, let's say, have a predictive model about disease severity, bulk is always the way to go. So because it's phenotype level and it's cheaper, it's more scalable, and also, well, I always have this discussion with also other people, so whether it's actually needed or not, and then they argue that connecting to clinical trials to see whether single cell would be useful for that, and whether you go into the clinic and just do, you get single cell RNA-seq for you, but that would be more expensive. So people say this would get cheaper, but the question is whether it can give you more information, at least for doctors, compared to liquid biopsies, compared to bulk RNA-seq, and so on. But I think in some cases, let's say in development, and subcellular resolution. So this is something that I actually showed in one of my papers, that there might be a subtle trajectory between disease and healthy. So in moderate, let's say in the case of COVID, so you have like, COVID is a super, I think is an easier case because the transcriptional response is strong, but you could see that like subpopulations that I don't think could be easily recovered with bulk. So I think it's just like case by case in development, and then if you also in clinical trials, and if you want to go and find a target at some point, then it might be needed to do single cell because you want to do cell type specific, just to be sure that you're on the right track. So, but as Shai said, it is basically complementary. Yeah.
Thanks. So actually, yeah, I think you're reaffirming some thoughts that I had. We did, and I think it's important in several different areas. So we did work over a decade ago on, this was still when we did gene regulation work in yeast, and we were measuring the cells in many different conditions at very high quality with promoter, GFP, fusions, and so on. So the expression data was at very high accuracy, and we could show that you move the yeast between different conditions. There's a lot of changes in gene expression, but they could be explained by very, very few genes. So 8 or 10 different genes could be predictive, like you said, of basically the entire transcriptome in that condition. And it also goes in the microbiome. We and most of the field are looking at DNA, but there's also metatranscriptomics, which is much more expensive because RNA are expressed at different quantities. So if you start to sequence, you'll first sequence the housekeeping genes of the most abundant species, only then the lower abundant genes in the high abundant species, and then you'll need a very, very deep sequencing to even begin to see things, and you don't need it because, in my view, because bacteria are typically found in one state which has some gene expression state. You don't even need to know that, because once you know the bacteria is there, it's deterministic to knowing the transcriptional state, like you said. So,
but what would be interesting is, like, I don't know what you and Shai probably think about is, like, in case of like small molecule screens. So we had this discussion actually as part of like grant writing with the reviewers, that someone said, like, why do you even need to do single cell for this? Because you just can straight away do bulk sequencing, and I think there are like two parts here. Small molecule, where you usually have like strong responses, so that's why potentially bulk would be super useful and cheaper. But on the other hand, then you have like CRISPR screens where you have super solid responses, and then solid cell states could be recovered, which might not be possible to recover with bulk. So I think that even like based on the type of screening that you do, then there might be selection here.
Just to say, it's not that I hate single cell. It has high value. So I think that you hit it, straight on the nail. That basically it's with trying to understand mechanistic intracellular regulatory expression, plays a role. But again, I think it's complementary.
Okay. Other questions from here, from the crowd? different topics to discuss.
Okay. In the era of new technology, everything is changing very fast. And my question is regarding the, for example, we have the virus, and after some times it changes and it has the mutation. And previously, for example, researchers had some, prescription how to deal with this virus or with this bacteria, and how they handle these mutated bacteria or viruses nowadays, because it's very fast. Everything is changing too fast, and we should how to handle this. Did you get my question? Did you get the question?
Can you try to repeat the question?
Okay. The viruses and bacteria, they have the mutation and they changing. Like nowadays, they changing very fast. And previously, we had the prescription, for example, for COVID. In the first period, we had the COVID-19, so on, and you had the prescription to deal with this. And in the second period, they had another mutation of COVID-19. It was difficult to handle. Previous prescription, it doesn't work. But with second, updated virus, how can we handle this? It's very difficult to find a good way to solve, to predict the disease or so on.
I think I can maybe try and give a hint on this. So if you look at viral evolution, for most strains, and the one I'm most familiar with is influenza. So influenza mutates rapidly, and we develop vaccines against it, and it keeps mutating. And if you look around the world in any given point of time, and depending on the season, you're going to see thousands of different strains, right? So from a World Health perspective, the problem has been, how do I choose what the vaccine would be? And this is something that existed way before our most recent mRNA vaccine kind of successes, which have allowed us to do rapid release of new versions of the vaccine. The trick has been that if you look at the kind of where computational biology has gone in, and it's been important, is that when you look at viral evolution, it's not a random mutation that you would think of. Actually, the mutations come in particular kind of hotspots on one hand, and versions of the strains that succeed usually have mutations in particular areas. So ultimately, the sort space of how I need to think about the next evolution of the next variant isn't as big as the entire universe that the virus could mutate into, right? So if you take this to thinking about a pandemic, you can start, once you understand the dynamics of how viruses evolve, you can start not be 100% sure, but you can start shrinking the space, right? And potentially this could also work for antibiotic resistance. I don't know. I'm not the expert here.
I think as well, one of the big game changers this time around has been the sharing of data and the extent to which different countries have released the sequences of the various variants. And I think it's the first time ever that we're seeing that level of collaboration. And I think that's the only way to ultimately deal with the velocity of evolving diseases.
You want to?
Yeah, maybe just to add, with antibiotics, I think it is very much like the picture that Shai described. It's interesting because mutations is a stochastic process, but to a large extent, under strong selection, it behaves almost deterministically. We can say with good probability what would be the next mutations, the next set of mutations that bacteria might take. Out of all the millions of possible changes, we can say, hey, it's only a change here and a change there that can actually give a substantial benefit under selection. And therefore, again, in some cases, not always, evolution becomes predictable in a sense. And I think that's is on one hand fascinating. On the other hand, it's kind of, it really connects us to, I think, the theme of that meeting, which is how computationally, with machine learning approaches, with AI methodologies, we can start predicting something about the evolution process itself, which, despite the fact that it's a ground is a stochastic process. So stochasticity plus strong selection sometimes behaves semi-deterministically and therefore in a semi-predictable way. Yeah.
Thank you. So today we've seen lots of, I think, amazing examples of science from the UK and Israel, but which are already very established scientific bases. For somewhere new here then that's trying to establish itself, such as in Abu Dhabi, can I ask each of you in your area, what opportunities might there be for an up-and-coming researcher in Abu Dhabi then to make contributions to your specific field of AI?
I'll give it a first shot. So what I talked about, there's another side to what I do in our research in the lab is explore the immune system and how it varies. Right now, I actually think that immune variation is one of the greatest challenges that exist kind of going forward as we think about an age of precision medicine. We think normally, we say precision medicine traditionally has been genetics, but really the immune system is all about the protection and so forth. And it greatly varies between us as individuals, and it varies as a function of demographics and environment and so forth. Exploration, so most of what we know about the immune system actually comes from places where, pharma companies have existed and kind of measurements have existed for many years. And what you basically see is, and I think the most extreme example I know is the one of many, rotavirus vaccinations. I kind of ran into this a few years ago. Rotavirus can be a devastating disease, kills children. In the US, 95% of children would respond to rotavirus vaccine. In Bangladesh, 50%. Okay? That's how bad our understanding of how we develop these vaccines. As we're going into this new era where science is shareable and measurements, as Orhan said earlier in this talk, we now have many different data types. I think a place like Abu Dhabi has really opportunity to lead, both from a data generation perspective, where I think this is, I kind of heard a bit about the genome project going on here, where there's probably a huge amount of capacity to generate data on one hand. On the other hand, this is an environment that's very different from where, we've studied human health to date, and understandings that could be derived here in Abu Dhabi about the immune system, but in other things I think about human variation, could have a lot of effects in kind of untapping healthcare that hasn't, that we don't have the information right now at all, so.
I'll add to that and strengthen it also for the type of work that we do. I think that in all of the models that we develop, you need some variation, right, in we always have machine learning, classic machine learning problems are you have your X variables and you have your response Y variable that you're trying to explain, and you need to see as much variation in the X to go with variation in the Y so that you can learn models that will generalize to more situations and more perturbations of the system. That's how you learn. So, so in that sense, exactly like Shai was saying, I think when we go and measure the population in Israel, Israel is actually considered to be a fairly good place in terms of a lot of genetic diversity, people coming from not too long ago from diverse origins, but still we're exploring a very tiny fraction of genetic, cultural, behavioral, environmental, climate, and so on. And I think in that sense, the Emirates is actually a place which is very different than Israel. And so I think that's why we're excited about now doing the type of work that we do in Israel, forming a cohort here in Abu Dhabi, in the Emirates in general. I think the capabilities to do this are very, very high here. Shai mentioned the Emirates Genome Project, which I think has been growing the fastest worldwide in terms of sequencing genomes. So there's infrastructures and capacity, and I think once we establish such cohorts here, like the UK Biobank, like the one we're building, doing parallel efforts here, I think that can add great value, especially when it's merged together with the other data sets that we have. So, yeah.
Maybe if I can add, I'm actually looking here at the mission statement of the university, and I'm especially attracted to the word global. And I think there's something very interesting about building a place that, whose mission is really to bring people globally from around the world. And that, I think, opens a lot in two ways. One, of course, is exchange, and I think that, in a way, is a lot of what happens here. But I think the other is very much the paradigm of some of the basic questions that we want to ask when we apply AI to data, to clinical data, is trying to see how different type of approaches, clinical approaches, affect overall clinical outcome, and especially this is important in questions that involve epidemiology and infectious disease. So, for example, if I'm thinking about the global community effect of prescribing antibiotics, and I'm looking in Israel, I would want to ask, hey, is there a different type of distribution of antibiotics given in one city in Tel Aviv and in another city in Haifa, and do we see prospective future effects of these different types? But the answer is no. Israel is pretty much, it's a homogeneous place when it comes to clinical practice. More or less the same antibiotics are prescribed by different clinicians in Israel because they follow similar protocols. Okay. But if I go to a different place, if I go to places in the US, and if I go to a place here, we may find, and I'm sure we'll find, that there's a whole different set and probabilities of using different drugs, and what is the effect of that different practice, clinical practice, on the accumulation of antibiotic resistance in the community is something you can really only start tackling when you compare different remote geographical places that have different clinical behaviors. So I think that really opens up a lot.
I think one other thing that would be super useful, specifically in the region that we are, is, and then the situation in the Emirate is intersection of like health and AI. I think there is a big, I think like a strong and tall wall between ML, AI scientists and biology. So actually lots of like hardest problems for ML are in biology, because anything that we are dealing with in biology is basically out of distribution, and then like because normal in biology is boring, so you always try to find out anomalies, and that's where the failure cases in ML happen, where you try to generalize to very different stuff. And I think what would be nice here is if there would be, let's say, kind of like, I don't know, like hackathons, problems, or data sandbox that could be the data could be generated here or somewhere else, and that could be provided as a sandbox where like AI, ML researches on impactful problem in biology. And that's where I think would be super useful here, as mentioned. And one other thing, because I personally have like roots here in Middle East, I think thinking about countries where they're underrepresented here and providing opportunities for those, and both on the data side, for example, like I don't think there would be like a Middle East atlas for single cell. There is nothing like that. I mean, no, because all the data and then just comes from like, you know, US and Europe, and then like the people who can actually do the data and stuff. And so there's a lack of data here. So there could be also consortia level efforts here on the data collection too. Yeah.
With your permission, I actually want to take the answer to a slightly different direction. And it's about the character of the researchers. So from the standpoint of like a small startup that tries to move fast, I believe the two things are extremely important to develop, like in terms of character. One of them is to really be bold. Like I think it's very easy like to follow existing paradigms and respond to like research paths that are dictated by someone else. And the second thing is the ability to kill ideas quickly. And that's like, that's really harsh, because you love your ideas and you think, exactly. And like I get the feeling that in life sciences related things, it's relatively more, like it's more difficult to fail, because you have like a lot of uncertainty and you always have some hope that things will work. But nevertheless, like even in a daily life, the ability to check a direction like quickly and be able to move fast and leave it behind, I think it's critical for people that will be able to really affect the industry down the road. That's my.
I don't really have much to add, but I like to just build on what others have said. I think that the heterogeneity that the region has to offer, along with the modern infrastructure and availability of funding, just really opens up a lot of opportunity. And so, yeah, I'd like to see, you know, startups, novel ideas getting funded and taken forward. I think also I'd like to see a lot of new technologies be deployed at the point of care. I think that would be a great thing that could come out of the region for sure.
Any other topics, questions? Okay, I think we've had a long and very fruitful day. Let's thank all the speakers again, and thank you. And look forward to tomorrow, another great day. Thank you.